You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech Podcast, and I have Dr. Andreas Werner. He's at the Institute for Cell and Molecular Biosciences at Newcastle University. So, Andreas, thanks for coming. How are you doing?
I'm very well, thank you, Richard. It's a pleasure to speak to you and tell you about my science and my interest.
Yeah, very good. So let's start there. What are you working on in particular as it relates to the genome, the human genome?
Right. So there are basically two things I'm working on. The first one is on natural antisense transcripts. That's in a larger context. That's a very interesting phenomenon. If you look at all the genes we have on our genome, you could basically kind of very neatly separate them out and have them regulated and controlled each individually. However, that's not the case. Genes are often clustered and they interact with each other. Transcription occurs in both directions, and that at the first sight that doesn't make sense, because it can happen all sorts of things. I mean, transcriptional machineries can bump into each other, or transcripts can be formed that hybridize to each other and form double-stranded RNA, and that is basically disastrous for a cell. And nevertheless, our genome has quite a number of natural antisense transcripts and areas that are transcribed from both directions and actually form these double-stranded RNA structures. And that's what I'm working on at the moment.
So you said some of the genes will interact with each other. Is that during transcription or just in general?
It's probably not right during transcription, because if you have two polymerase complexes moving towards each other, they interfere to a certain extent, and you can show that very neatly in bacteria or in yeast. And if you put two promoters up and downstream of a gene and let them both go, and the transcripts interfere, and basically they don't happen both at the same time. However, in a high eukaryotic genome, it's probably transcription occurs not kind of switched on and then it runs and runs and runs and runs, but it's kind of transcriptional burst, and so it's rather an intermittent transcription that can be silent to a certain extent, and then the opposite transcription could take place. So it may happen at the same time, but just kind of in an intermittent fashion.
If you have a given sequence of base pairs, let's say it's 1,000 base pairs long, then there's some kind of break, and then there's more base pairs. Could you read the 1,000 base pairs, but also read, let's say, the second 500 of them plus another 500 of the next sequence and create a different protein? Is the DNA readable like that, where you don't have to only read it at certain spots, but you can take parts of areas that we would call a gene and overlap with another area and create proteins out of that transcription?
Absolutely, yes, absolutely. However, you have kind of very specific. This is regulated in a very precise time and locus-specific or organ-specific, cell-specific way. So under normal circumstances, there is very little of this antisense transcription going on. And secondly, it's probably a very limited number of genes that actually do that. And you have to separate very clearly that one where this antisense transcription occurs, basically the biological locus or the biological organ where that happens is the testes, in male testes. And this is probably in a context of an evolutionary control. However, you have, and that has only occurred or being recognized in the recent maybe year, 2 years, you have a lot of this antisense transcription and double-stranded RNA formation in mitochondria, from the mitochondrial genome, and also under pathological circumstances from repetitive areas. So that's the main sources of this double-stranded RNA that occurs.
So what are you, in specific, what are you studying? What are you hoping to figure out?
The hypothesis we are currently investigating is that in the context of a cell that forms these double-stranded RNAs, these almost look like a virus. And the cell has to distinguish whether that's actually a cell structure or whether it's a viral structure. And if it's if it's recognized as a viral structure, it's dealt like a virus, and it triggers an immune response, an innate immune response. There are a number of proteins within the cell that recognizes these double-stranded RNA structures and then trigger an interferon response and an immune response, and in the worst case, trigger apoptosis. So it's a very fine balance within the cell to actually keep these double-stranded RNAs either very specifically localized within either the nucleus or the mitochondrion, or alternatively have it quickly recognized and degraded. And if that control mechanism breaks, the cell has a problem. And now the hypothesis is that possibly these double-stranded RNA could be a sign of a low-level stress that occurs maybe during aging, or, as I said, low-level stress, and then generate an immune response that could be a preparation for a pathological development like neurodegenerative disease or maybe cancer.
Well, how do you know, when does this happen? Is there transcription and then the RNA folds in upon itself to become double-stranded, or how does it happen?
Right. The two examples that I mentioned, the repetitive sequences and the mitochondrial DNA, that's kind of, in the mitochondrion you have both strands transcribed, that's for sure. The repetitive sequences in the nucleus are kind of, from a structural perspective, either they are transcribed in both directions, or they are repetitive sequences that fold back maybe on each other and form double-stranded RNA. However, under both circumstances, these in the mitochondrion and in the nucleus, these structures are confined. They are not leaving the mitochondria or the nucleus. And if they do, as I said, they are quickly recognized and degraded. So when you, for example, by adding a specific drug that induces transcription, reduces DNA methylation or changes the histone code so that you have a more permissive transcription, if you do that, there is a chance that the intrinsic control system is overpowered, and you end up with an increase in double-stranded RNA and with an immune response triggered by these kind of relaxed transcriptional events. And
you've seen that there typically is an immune response to cause the entire cell to go through apoptosis, or what have you seen?
In the worst case, yes.
What about in non-worst case? Does it simply call it a dud transcription and just I don't know, there's like a T cell engulf the RNA once it leaves the cell, or what happens?
So these events have been recognized because the cells have been stressed, either treated with drugs or specific protective mechanisms have been suppressed, so that the cells start accumulating these double-stranded RNAs, and under normal circumstances, this is not the case. There are enzymes like ADAR or PKR or also Dicer and that recognize these structures and degrade them. And you have to knock out some of these protective mechanisms to actually see then a immune response and at the worst case apoptosis. And if you kind of extrapolate that towards disease, many cancers and many neurodegenerative diseases, they have an underlying inflammatory phenotype, and that could come from these double-stranded RNAs.
So what is your goal? Is to understand how this happens and then the consequences of it, or is it to somehow intervene?
I mean, the consequences in a kind of an artificial, in an experimental system, are relatively straightforwardly accessible. However, in kind of an in vivo situation, it's very difficult to assess because the intrinsic double-stranded RNAs, they are at such a low level that without massive amplification, you can't actually demonstrate those. And as a consequence, the tools at the moment are not really established to have that kind of amplification or that specificity that we can find these double-stranded RNAs and distinguish whether they are confined within the mitochondria and the nucleus, or whether they leak out and actually are interfering with the cellular protective mechanisms like PKR and Dicer and these ADAR, these double-stranded RNA recognizing proteins.
So you can observe this in a lab, but you haven't observed it in vivo because you think it's a very rare event?
That's correct. Under normal circumstances, it's very hard. You may see it with a specific antibody, but right only within the mitochondria. And you have a number of protective mechanisms that prevent those double-stranded RNA from actually exiting the mitochondria. However, if you knock out specific protective mechanisms, then you can see that, and you can monitor or read out an inflammatory response, an interferon response, for example, on certain cytokines that are representative for an immune response. But only if you reduce the protective mechanism within cells.
So how do you know that this occurs at all? Because you're saying it occurs only when you have to knock out protective mechanisms?
Yes, that's a chicken and egg question. You knock something out, and then you trigger a specific response that you can also trigger by infecting a cell with a virus. So that's basically
I was going to ask you why this happens. So this is this happen. Viruses can trigger this to happen?
Yes. Originally, all these defense mechanisms, they have been described as a response to a viral infection, and the very key experiment is to transfect cells with a double-stranded RNA construct, and then measure the readout, which is an inflammatory response. And then you can trigger the same inflammatory response with the same characteristics when you, for example, reduce reduce DNA methylation, increase transcription of repetitive elements, or when you promote a leakiness of mitochondria, so that mitochondrial double-stranded RNA leaks out into the cytosol. So that's the viral response is actually the first step of characterizing this response. And I think the new thing here with that is that it's a very kind of a rare event and something like a very tuned machine that is functioning very well, but then when it gets old or when it gets stressed and the protective mechanisms don't work as well as they do, kind of eventually this double-stranded RNA builds up and eventually kind of breaks the camel's neck, and eventually an immune response is being triggered. So it's a very slow process.
What's the benefit of this happening? Is there any benefit to the organism besides an alert that something is wrong, or is there another benefit?
That's a very interesting question. I think in biology you often have a benefit on the one side, on the one hand, and an absolute catastrophe on the other. And biology manages quite carefully to meander between the two. And in very few cases it comes to an absolute catastrophe, but in probably in experimental cases you can trigger that. So this issue with double-stranded RNA that has only arisen about 2 years ago, I would say, or 2 or 3 years ago, that has become a hot topic. Before that, there was kind of circumstantial evidence that you have antisense transcripts. So all these genomic assessments of the human genome, etc., they have shown that loads of antisense transcripts are being transcribed. They potentially form double-stranded RNA, and they are most likely or most predominantly expressed in testes. So most likely what we know so far is that a beneficial effect of having these antisense transcripts, also having the repetitive elements that can, if kind of released in a brain cell or so, have detrimental effects. In testes, there are regulated events that kind of release those these repetitive elements, and that has to be seen in an evolutionary context, so that these repetitive elements actually contribute to generate variability, novelty in a sperm cells. And because we produce so many sperm cells, we can afterwards deal with that variation that in most of the cases is detrimental, so we can just sort them out. And the hypothesis is that these double-stranded RNAs help to assess and to scrutinize the genome after all these kind of events have happened that create variability, that are basically a playing field for evolution.
Well, I would think that the repetitive sequences are there for redundancy and to make sure all the systems are running well. But you said they actually could be a source of variation?
Yes, true. I mean, again, these repetitive sequences, we have them, and what do they do? I mean, there's no obvious sense for having a vast majority. I mean, more than 50% of our genome is just kind of repetitive sequences that don't code for proteins. They don't have a biological function. And however, if you look at it, the way that these repetitive sequences are actually new material to generate new genes, then you have a huge resource on the one hand of genetic material. That's a very rare event. But in addition to that, these repetitive sequences, they have regulatory power. So they have transcription and enhancer activities. And when they kind of reintegrate somewhere, they may influence the expression of the neighboring genes. And that's probably another way of how these repetitive sequences actually can interfere and create novelty within a genome. Again, that happens at the very low incidence, at the very low level, but it is a very important step within human sperm development, for example, and higher eukaryotic sperm development, that these repetitive elements actually have a window of retransposition and of activity.
And you're saying that this happens predominantly in the mitochondrial DNA and in and around the mitochondrion, or does it happen in the nuclear DNA?
I apologize. I probably explained that not very well.
Okay.
I have to separate the two. So the mitochondria, they form double-stranded RNA, and that's a consequence of how mitochondrial DNA is transcribed, and that's very, very different from the nuclear situation. As a byproduct, they produce double-stranded RNA, which I don't really don't see a reason or a benefit of creating that double-stranded RNA, but it happens. And the only reason why this mitochondrial double-stranded RNA kind of came into focus is because it is there, and if it leaks out, it triggers this immune response, and may feed into an inflammatory response. The reason I ask is that I guess supposedly mitochondrial DNA has passed through the mother, not through the father. So if this happens to a woman, I wonder if it affects her epigenetically, and if that gets passed on, especially because, like I said, her mitochondrial DNA get passed on to the offspring, versus if it happens in a man. That's why I thought about that. That's an interesting point. I don't think that's known. How the inheritance of the double-stranded RNA. I don't think that's.
And also too, like, I don't know, the sperm are produced all the time, but eggs, I mean, supposedly they're, a woman is born with all her eggs, and I mean, I've recently read that she may produce more, but I wonder if the consequence of that is it happen more in men because there's more cell divisions and more creations of new cells, the spermatogenesis, or is it less likely to happen in women because they undergo, it's the same cells that are just aged, so it didn't happen when they first were formed, so it doesn't happen after?
Yeah, I think you're absolutely right there. That because the finite number of egg cells are available, there is very little room for variation there, because with a number of offspring that is what is it, 1, 2, 3, maybe 5, or in the old times 10, but certainly not the numbers of bacteria or so of lower complex organisms. The number of offspring they generate is gigantic. The quality of reproduction has to be absolutely exquisite to produce something. You have 1 or 2 goes, and that's it. So basically, the whole question of variation and of creating something new has been outsourced towards sperms, because we produce billions of sperms, and that allows us to kind of exploit or have a limited kind of playing field for variation. And that's probably why sperm cells have these demethylation phase and higher variability as compared to eggs.
Okay. Have you seen that? Is there any permanent creation of double-stranded RNA? I mean, is this something that can be modulated by epigenetic changes and stay permanent, or even be heritable?
I don't think I perceive double-stranded RNA in the evolutionary context as a tool, and not necessarily as a messenger. It's a tool to distinguish whether something is transcribed correctly and folded correctly. It's a tool to scrutinize the quality of the genome after recombination, after retransposition of transposon, etc. And I don't perceive it as an agent for inheritance to be passed on.
I guess it's never a beneficial effect. It's always negative, right?
No, I think you can't put it in a way like that. I think if double-stranded RNA was only negative, nature would have selected against it, and that's clearly not the case. Then we would have arranged our gene very nicely, kind of split from each other and separated, and never converge in transcription or so that could potentially give rise to double-stranded RNA, and that's clearly not the case because rather the contrary, we tend it's not a very clear tendance, but it's a tendance that we rather have more double-stranded RNA and gene loci that are transcribed in both directions that potentially form double-stranded RNA, that this is actually a somehow beneficial arrangement of genes.
So have you studied this in human models or mouse models, or have you seen the appearance of this in multiple creatures?
In all sorts. We do research in mouse, and we do that. We look particularly in testes because there we think that's where the natural situation, the biology happens for double-stranded RNA formation and for a potential regulatory or a potential biological role for double-stranded RNA that is in sperm. So we do that in mouse. We look in cell cultures and on a larger genomic basis, but interventions we do in cell cultures. And this is very, very difficult because if you look at two genes or a gene that is transcribed in the opposite direction of another one, and you have two RNAs that potentially interfere with each other, you take a model system and you start either overexpressing one of the components, you can bet that the other one is either downregulated or stabilized, and you knock out the other one, and you can bet that something happens to the partner RNA as well. So it's a very difficult model to actually investigate on a mechanistic basis.
Gotcha. So what do you see as like the near-term future of your work? What are you hoping to elucidate maybe in the next year or so?
Within the next year, I would hope that we can find a very sensitive assay that we can find double-stranded RNA at a low level in cellular location where it doesn't belong to, so not in the mitochondria or not in the nucleus, but in the cytoplasm. And that we can demonstrate that, find that, hopefully even quantify that, and find a predictor for diseases, for degenerative diseases.
Do you know of anyone that's been able to make a quick question? Do you know of anyone that's been able to make a tiny sensor that can be put inside a cell's cytoplasm to monitor one cell? Has anyone ever done that?
I would not like. Well, yes, a sensor. There's an antibody that recognizes double-stranded RNA, so we're going to use that. But you can also use potentially protein or protein fragments that recognize double-stranded RNA, so that you probably need a parallel detection or kind of an enforcement between two methods of detection to actually be specific enough. So yes, that would probably kind of come close to a sensor that you put into a cell. I mean, then after, it's obviously the question is, how do you, if you look at brain cells, I mean, it's not really feasible that you test an in vivo brain cell of a patient. So this kind of degenerative process also happening in other cells that are better accessible as compared to brain cells, for example. That's a big challenge. So, but
okay,
I think
I kind of interrupted you. So over the next year, again, what do you see yourself being able to accomplish to move the research forward?
I think right. So on the biological side, in testes, we hope to have a clearer idea of in which genomic loci these double-stranded RNAs are formed. under what circumstances, and hopefully have a better understanding on a natural basis what these antisense transcripts are good for, and the double-stranded RNA that is formed between the antisense transcript and the sense transcript. With respect to directly monitoring double-stranded RNA in somatic cells, as I said, I hope towards probably having a method to, at least in a cell culture system, to stress cells, maybe age cells, and then show that double-stranded RNA actually plays a role in old or stressed cells and is a potential readout for the level of stress or the level of aging, the progression of aging that happens in these cells.
All right. Well, very good. Any final thoughts or things that you want to express? I feel like I asked you some tough questions. So any?
That was. I mean, I think it's since I started with RNA. I mean, when I first discovered these antisense transcripts and kind of unusual transcriptional events, that was in 1995 or so. So there weren't any genome sequenced, and I just thought, well, if a gene is transcribed in both directions, and this is actually quite unbelievable, and if a cell does that, there must be a reason. And I started studying this, and then came the human genome sequence, and suddenly people reported antisense transcripts all over the place, and noncoding RNAs, etc. And it has been a whole revelation and a whole revolution in new concepts and absolutely new findings. And I think it's absolutely brilliant that after a time where you had the impression that, well, I mean, we basically know what happens, we know the genes, etc., how a cell works, etc., that basically we have a complete overhaul of a perception, what the gene is, how a gene is regulated, how RNA works, etc. And it's absolutely fascinating how new aspects of this whole RNA world and keep popping up, and it's a never-ending story. And I look forward to further exciting new developments, new discoveries.
Well, that's great. Well, Andy, I appreciate you coming. What's the best way for folks to get in touch or to find out more about your work?
You can always write me an email. That's probably the best way to contact me. My email address is at the University of Newcastle. You'll find it when you find me via Google. It's might be easy if you Google my name and Andy, and if you add tango, Argentina and tango to it. That's my second hobby. Then cool. I'll pop up. Do you want me to spell out my email address?
Yes, please, if you don't mind. Yeah.
Oh, okay, that's Andreas.Werner. That's WERNER at ncl.ac.uk. And I'm more than happy to reply and discuss and get in contact with everyone who is interested. And please do contact me and give feedback and tell me what you think about these interesting double-stranded RNAs.
Excellent. Well, Andy, thank you for coming on the podcast. I appreciate it.
It's it's my pleasure, and I hope that whole initiative is a success.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.